Recently, Yale University's research team has developed a new cancer immunotherapy that uses CRISPR technology to activate multiple cancer-related endogenous genes to help the body's immune system recognize, attack and kill cancer cells. Preliminary results indicate that the CRISPR activation, CRISPRa, platform-based multiple activation of endogenous genes as immunotherapy can effectively kill or eliminate many types of cancer in mice. On the 14th of October, the results of the study were published in the journal Nature Immunology, entitled Multiplexed Activation of Endogenous Genes by CRISPR Elicits Potent Antitumor Immunity. In recent years, immunotherapy against cancer by the body's own immune system has revolutionized the development of cancer treatment. Immunotherapy can recognize abnormal peptides produced by the expression of cancer cell mutant genes and label cells as non-self or foreign matter, triggering an immune attack. Currently, immunotherapy mainly includes immunological checkpoint inhibition, adoptive cell therapy, human recombinant cytokines, non-specific immunotherapy, oncolytic virus therapy, and cancer vaccine. But all of these treatments face various limitations, not foolproof, and cancer cells can escape the check of immune cells. Tumor-associated antigen, TAA, is produced by mutations in cancer genes, and recognizing TAA is an important step in cancer immunotherapy. One of the key issues is that the level of expression of cancer gene mutations may not be sufficient to elicit an effective immune response, and cancer cells often downregulate the expression and presentation of antigens to evade immune recognition. To address the immune evasion problem of cancer, the research team led by C.D. Chen of Yale University developed a technology that combines adeno-associated virus, AAV, gene therapy and CRISPR gene editing, MIGI. Unlike previous immunotherapy, the MIGI method does not search for and edit target DNA sites and insert new genes. Instead, it targets cancer-associated genes of specific cancer cell types, allowing the immune system to attack markers by pinpointing, labeling, and amplifying signals cancer cell. First, the research team used CRISPR activation technology to combine catalytically inactivated Cas9, dCas9, with a single guide RNA, sgRNA, to target multiple genes, regulate thousands of cancer-specific gene expression and antigen presentation, and enhance immunity, system identification and lethality. At the same time, the researchers have upgraded the MIGI system several times, such as the genome-wide AAVG MIGI. Animal experiments showed that whole genome-activated tumors were completely eliminated in more than 80% of mice, and this tumor clearance was mediated by acquired immunity. Thus, AAVG MIGI can induce an adaptive immune response in a host against a variety of invasive cancer types. Subsequently, the research team developed an upgraded version of the AAVG MIGI system, namely AAVP MIGI. Through whole exon sequencing, the researchers identified all the mutated genes in the E0771 triple-negative breast cancer cell line and designed the MIGI CRISPR precision activation library that targets the tumor rather than normal breast tissue. Thus, the AAVP MIGI system targets more than 1,000 gene mutations in E0771 breast cancer cells, triple-negative breast cancer. The results showed that 44% of animals with triple-negative breast cancer showed near-complete response, NCR, or complete response, CR, after AAVP MIGI treatment. Excitingly, in the first 3 months after tumor transplantation, all 9 animals showing CR can still completely eliminate the same tumor attack, and these animals can achieve long-term complete remission for more than 180 days, indicating AAVP MIGI induces an effective, long-lasting anti-cancer response. At the same time, the research team also tested AAVP MIGI in mice carrying local and distant tumors. The anti-cancer response of local tumors and distant tumors, without AAVP MIGI treatment, was observed simultaneously after treatment of AAVP MIGI with local tumors. After treatment with AAVP MIGI, the CR rate of distant tumors was 67%. By single-cell sequencing analysis of the immune cell population in the tumor microenvironment, the results show that MIGI can effectively increase the expression of target genes and antigen presentation, enhance the effector function of T cells, and recruit CD4+ and CD8+ T cells to tumors. microenvironment that promotes local and systemic anti-tumor T-cell responses, including triple-negative breast cancer, melanoma, and pancreatic cancer. The author of the article, Dr. C.D. Chen, Department of Genetics, Yale University School of Medicine, said, enhancing the expression of endogenous genes in cancer tissues can amplify the non-self characteristics of cancer cells and mark them as targets for immune attack. MIGI is a kind of the new immunotherapy can effectively transform cold tumors into hot tumors, help to recruit effector T cells to tumors and reshape the tumor microenvironment, enhance T cell infiltration and anti-tumor immunity. This treatment strategy is like the cancer cells are marked with a very prominent mark, and the immune system can quickly detect them and launch an attack. The latest study, published in Nature Immunology, demonstrates that activation of endogenous mutant genes by CRISPR activation technology can amplify specific signals from cancer cells and induce effective adaptive anti-tumor immunity. Multiple endogenous gene activation methods can accurately target multiple genes or even entire cancer genomes without harming the genome, is a versatile and highly scalable strategy, or is effective against a variety of cancer types, including current resistance to immunotherapy cancer. As a potential tumor virus gene therapy, AAVP-MIGI can be used for surgical adjuvant therapy of primary tumors, metastases or residual lesions. More importantly, when AAVP-MIGI activates the endogenous gene, the tumor acquires additional mutations that remain the target of MIGI. Next, the research team will continue to optimize the method to prepare for manufacturing and cancer clinical trials.